Every person's immune system harbors a wealth of information about their lifetime of environmental exposures to things such as allergens and microbes, and even whether diseases like cancer or autoimmunity may be developing unnoticed. To extract this valuable information, scientists at Johns Hopkins and Harvard have developed a high-throughput blood test and an antibody analysis platform called PhIP-seq. By bringing together customized DNA synthesis, phage immunoprecipitation, and next-generation DNA sequencing, scientists can now identify the molecular targets of past and ongoing immune responses. Customized DNA synthesis. The process begins by designing hundreds of thousands of DNA sequences to encode collections of desired protein fragments, for example, all known allergens or all known viruses. The DNA sequences are then chemically synthesized as an array on a solid surface and altogether released into solution in a single tube for subsequent cloning into the genome of a bacteriophage, a virus that infects bacteria. T7 bacteriophage display. As the phage library replicates and assembles in bacteria, the protein fragments encoded by the cloned DNA sequences are expressed on the surface of the phage capsids. This system, which physically links DNA sequences to their encoded protein fragments, is called phage display. This phage library is then loaded into the multi-well plates that permits parallel analysis of large numbers of individual samples. Phage immunoprecipitation. A very small amount of blood containing antibodies is mixed with the phage display library. The antibodies from the blood find and bind to the protein targets they recognize. Magnetic beads are then used to capture the antibodies along with the phage particles they've bound. A magnet is used to separate bound from unbound phage. Phage particles that display protein fragments not bound by antibodies are washed away at this point. Next-generation sequencing. The DNA sequence within the captured phage particles are amplified by polymerase chain reaction, or PCR, a process which is also used to label each sequence with a sample-specific DNA barcode. The PCR products from the multitude of samples are mixed together and analyzed on a high-throughput DNA sequencing instrument. Each instrument run produces massive amounts of antibody binding data for each sample. The data are then run through an informatics pipeline to produce multiple types of analysis. At the individual level, PhIP-seq can be used to detect viral infections, allergies, and autoimmune diseases, for example. When applied to defined groups of individuals, this technology can help scientists better understand the essential role of the immune system in complex diseases, such as autoimmunity, asthma, neurodegeneration, and cancer. PhIP-seq is therefore an exciting new technology being utilized by biomedical researchers in many fields.